When I saw this article concerning bioplastic, that caught my curiosity because I was like, how is biology playing a role in the production of plastic? And then it followed up with velvets. Velvets are quite an interesting creatures. They are pretty much worms, and based on how they are described, one in terms of the ancestry of velvet worms, they've been at least around 200 million years, and this here is based on fossil records. And thinking about the size of velvet in terms of the post-Cambrian era, it's pretty much the same size approximately as the ones that you will find currently in Costa Rica. And there are a lot of studies that are based on this particular type of species from Costa Rica, which is pretty diverse and awesome to see how different research methods are similar and how they actually come up with conclusions based on their findings. This here, in terms of the location of these velvet worms, comes across pretty much around the tropical and the temperate forests, and based on the anatomy of these velvets, you have the dozen legs, the size of a pocket, pretty much around half an inch to close to 8 inches long. And looking at how their sight is built, they have limited eyesight. That means that they really sense their surroundings based on the bumps that they come across along the way. Now, in terms of their facial setup, they do have nozzles on both sides of their face, and so this here now integrates it into the behavior, how they behave based on their surroundings. In terms of the hierarchy of the food chain, they are considered to be territorial based on the type of bugs they eat. They eat a lot of crickets and they eat a lot of woodworms. And this here, they capture their prey by shooting sticky slimes at their prey. And this sticky slime is pretty much impressive in terms of how its characteristics is portrayed based on the strategy that it does to species. And this slime here is all comes down to pretty much 4 steps. The first step is that once it is in the glands of the slime, no, of the velvet, they are pretty much watery liquid in its state. But as soon as it comes out, in mid-air, as it is sprayed over the prey and sticky to the ground, it looks like a jelly-like rope, and this here then transforms to a fibrous strand. The reason why that transformation comes to play is because of how the prey tends to fight it off. However, that improves the strength of that particular liquid slime, and so this here then leads us to the final phase, which makes it to be look like pretty much a glass like solid. This here is hardened within seconds, no doubt at all. And so this particular analysis here is based on Matthew Harrington's at McGill Biochemistry department. And so this particular research group, their experiment is to get this particular slime, study the chemical reactions that is making it to go from those particular phase, and how this can be potent in terms of our production of various plastics that may be important in the future in terms of better chemistry. But the one thing based on the history of this research comes down to, first, in the 1870s, they actually discovered this particular characteristic of this slime to be bitter in terms of the taste. I don't know how someone thought it was a great idea to test it out, but hey, I'm glad it didn't die. They survived, and they figured that it actually had a bitter taste, which it does currently now. In terms of looking at this particular liquid, how it reacts with various elements, it says here that when you soak this glass like solid in water, this reverses the process to get it back to the water liquid phase. On the flip side, this robbery by rubbing the water liquid phase, you can forward the process to get more of a strong fiber like nylon. So, looking at these two processes, the forward and the reverse reaction, they actually came up with the notion that the fibrous formation is due to the behavior of the proteins that are present in this slime goo that is in there. Now, what is the goal of this particular experiment by this particular group? The goal here is to isolate the protein out of this slime glue, and thinking of the difficulty level, this here is from moderate to quite difficult in terms of how to extract the protein. And you might be asking yourself the reason why. Why is it difficult to get this protein out? The process is pretty much due to the physical condition of the slime glue, as you can see from how it transitions from one state to another. The phase shift is really instantaneous, close to instantaneous, going from just the liquidy, watery state to then heading into the solid, glassy like state. All comes down to pressure. Either you are pipetting or just transferring it from one medium to another. That is a lot of pressure that is quite sensitive by this particular goo, and so in order for them to overcome this particular problem here, what they had to do was generate the RNA sequence for that specific protein, but they needed a decent amount of number 3 species that are in that particular velvet worm family, and the 3 locations that these 3 species were located in are Barbados, Singapore, and that of Australia. And so the next phase was, after they got the RNA sequence, they just put the RNA sequencing into a particular program called the AlphaFold 3. And what this does is that it uses AI to give a prediction as to the quaternary, let's put it that way, shape of this particular protein. And you probably just ask yourself the question, what is the result of this particular protein structure? The result that this AlphaFold 3 brought up was that, hey, the shape is more of a horseshoe shape. And looking at also the content of the amino acid, the most dominant amino acid in this particular protein is leucine rich, and leucine is known to be a non-polar, which is highly needed for protein synthesis, muscle health, and that of metabolic mechanisms or regulations. And so this here has brought up the question as to how can we have some parallels between this particular protein and something that has already been in existence. And so this here comes down to some evolutionary connection, and this particular protein is quite similar to the toll-like receptor found in ancient immune system plants, vertebrates and invertebrates. And thinking about the location of this particular receptor is mostly at the surface of immune cells, and the function of this is to bind strongly to pieces of invading microbes and later let them go. And so based on this particular description of the toll-like receptor, it's kind of is parallel with that of the horseshoe-shaped protein function, because this performs a particular type of mechanism known as the host-guest dynamics, in terms of how the proteins that are grabbed together due to the help of the host protein can allow our liquid, the fluid slime, to become more of a fibrous because of the collection of all these proteins. But here is the downside of this particular ingredients of this particular slime. The downside is that we don't really know the specific proteins that are responsible for the toughness and also the elasticity of this slime goo. And so what we are looking at in terms of the future of this particular slime goo by the Velvet is that they need to understand the phase changes of the sticky slime goo, and this here will give us a lot of insights into better recycling techniques that are going to be implemented into bioplastics and also the plastics that are currently out there. Now, another future ambition of this particular research is that they need to develop important, replacements for plastics that can be broken down easily and reinforced into new shapes. And the next one here also is that they need to be able to investigate other proteins that are responsible for the toughness or elasticity and the mechanism by which they can interact with one another. And this is according to Yandrai Coleras Urena from Costa Rica National Laboratories of Nanotechnology. And the next step that this particular group, the Harrison team, are going to be working on is trying to purify the horseshoe-like protein and confirming its structure using electron microscope. So I hope you found this really informative concerning this incredible product that is produced by the Velvet Worm and how it's changing our understanding about better ways to explore the chemistry behind recycling processes of plastics, but at the same time looking at efficient ways that doesn't necessarily have to do with proteins, but studying how proteins can have a foundation for something that is much more economically friendly and environmentally friendly to recycle plastics. So I hope you found this really informative. Please hit the comment down below and let me know your thoughts about this. Do you think that they should, make this particular protein in huge amounts and have this be a model for recycling plastics or changing plastics from one particular shape to another? Or do you think that they should develop some kind of an interesting enzyme, let's just say enzyme or catalyst, that kind of have the same model as this protein and follow through with the similar chemistry that will lead to the recycling effectively of plastics that are currently around our environment that are not used or that need to be reused. I hope you found this really informative. See you in the next video. Stay smart as always and believe in yourselves.